patients and saves life. The goal of studying cancer is to develop safe and effective methods to screen or detect or treat or cure all disease or collection of disease called cancer, and even the cancer-related issues such as supportive care in oncology or the related emergencies in oncology field. So the better we understand those diseases, or the better we understand the cancer nature, the more progress we will make. Research help us to accumulate extensive knowledge. So we can, the more we understand the cancer nature, about the biological process regarding the cancer cell growth or spread or metastasis, the more knowledge we have to fight the cancer through the research weapon. Those discoveries have led to more effective and targeted treatments and prevention strategies. Breakthroughs in preventions or any cancer approach or discipline may take decades or even generations. I mean, it can take more long time, from in average 5 years to maximum 15 years or 20 years. So the wide array of disciplines may be over decades or even generations. So cancer research requires partnerships between several branches of science and several branches involving researchers, clinicians, patients, and others to translate yesterday's discoveries to today's advances. Research progress is not often linear, such as not related to the problem and the results. This is not the research. It is cyclical. The problem, so we can observe and then apply in the lab, then apply in medical field, then lead it to human application, and ongoing till we have a good knowledge about it, so we can change the practice. Advances are a result of constantly building and on earlier discoveries. The research is like, I would like to say, it's like a wall. We build it brick by brick. The brick put yesterday, so we will come and put 2 bricks today, and tomorrow someone who will go and put 3 or more bricks to build the advances. So the advances, like the research, is not often linear, but it's ongoing, and advances are more and more become clear to us throughout the continuous knowledge. This figure, I think it most related to cancer research. The roots are different basic science, such as pharmacology, biochemistry, omics, epigenetics, economic physics, stuff like this. So through that and the cancer research process, we can gain the fruits like screening, new screening approach, new quality of life of patients, healthcare organization gains, diagnostics, or treatment plans. So I think this cancer research is like a tree with more roots collaborate to give us fruit throughout our approaches, different approaches throughout screening or diagnostic or treatment or any other approaches of oncology treatment. Cancer research cycle. The cancer research, we stated before, it is not a linear process, it's a cycle. the cycle flow from observation with medical relevance through the patient's bedsides and back to lab. Progress in cancer research depends on participation and a harmony between the population scientists, physician scientists, and the clinical research, or even the healthcare givers, caregivers, and the healthcare providers. Insight from discipline can give us a light to the other disciplines. And the discoveries made in one cancer can offer new ideas to better address others. So the cancer research is continuous cycle. The cancer research started with all of this. Start all with the medical observation or the medical relevance, then the discovery, then the therapeutic developments, then the clinical testing. If the results are practice changing, so we can move it to the clinical practice and learning healthcare system. We start phase one, starting phase one and phase two and phase three, then submission for FDA to approval, then FDA review and confirm safety and effectiveness, then phase four as a market research. The clinical trial process is a long process from 5 to 15 years. Special designation can speed up this process regarding some medications, such as the compassionate use medication and the medication for rare disease or for accelerated use to help someone with a rare case of cancer or rare disease of cancer. So some special designation can speed up this process, and the special designation are well known with FDA and the EMEA, the European Society that equivalent to FDA. Now we will talk about an insight for oncology clinical research regarding the Destiny Breast phase 3. The Destiny Breast phase 3 is regarding the comparison of trastuzumab deruxtecan versus trastuzumab emtansine in previously HER2 metastatic breast, previously treated HER2 metastatic breast cancer. I choose this trial to show or give an insight to oncology clinical research. The both drugs are approved by FDA. The both drugs are used in treatment of breast cancer, and the both drugs in this trial will be used head-to-head in certain circumstances to treat the metastatic breast cancer. And we will see the change that will be happen in the practice. First of all, I will talk about some background. Antibody drug conjugates had shown promising efficacy in previously treated HER2 metastatic breast cancer regarding the trastuzumab emtansine or trastuzumab deruxtecan. Trastuzumab emtansine, which abbreviated here to TDM1, established at second-line standard of care based on trial called EMILIA, with median progress-free survival outcome from 6 to 10 months improvement. Trastuzumab deruxtecan, which abbreviated as TDXD, has higher drug-antibody ratio, 8 to 1, compared to TDM1, 3.5 to 1. So it can be more effective in inhibiting the topoisomerase 1 rather than other microtubules. Trastuzumab deruxtecan is FDA approved for treatment of adult patients with unresectable or metastatic HER2 breast cancer. Please concentrate with me, who have received 2 or more previous anti-HER2 based regimen, 2 or more in metastatic breast cancer, and based on anti-tumor activity in the single-arm phase 2 this Dynasty Breast 01 study. So these results resulted from phase 2 study, such as we mentioned in the previous slides. DESTINY-Breast3 is first randomized study to TDXD in head-to-head with TDM1 in previously treated metastatic breast cancer. And we will see the results. But before, we will start with the study design. The study design is randomized, multi-center, open-label phase 3 studies, stratification by HR status, prior treatment with pertuzumab, history of visceral disease, such as pre-mets or stuff like this. So patient with unresectable metastatic HER2 breast cancer previously treated with trastuzumab plus taxanes in advanced metastatic settings. So the randomized occur, and we give nearly 260 patients trastuzumab deruxtecan with dose 5.4 milligram per kg Q3 weeks, and the other arm 263 with trastuzumab emtansine. Medium flow-up was deruxtecan as 16 months, and trastuzumab emtansine was 15 months. Primary endpoint, the progression-free survival. The secondary endpoint was overall survival and overall relative response and degree of response and safety and drug-related adverse events. First, baseline characteristics, median age was range in nearly 54 years. Females, the regions between Europe and Asia and North America, HER2 status +3 as we need proven HER2 positive, performance status 0 or 1, hormone receptor positive mainly, visceral disease mainly, prior lines of therapy 0, 1, 2, 3, 4, and prior treatment of metastatic breast cancer, mainly prior cancer therapy with anti-HER2 therapies such as trastuzumab or pertuzumab, or anti-HER2 TKIs such as lapatinib, or other anti-HER2 antibody or ADC adotrastuzumab. First, primary endpoints, the progression-free survival. We can see that the median progression-free survival here was 12 months progression-free survival is highly statistically significant in favor of trastuzumab deruxtecan, with near 75% versus 34.5%. With hazard ratio of 0.28 and 95 confidence interval. The secondary endpoints, the progression-free survival by investigators in the multi-center, in the several centers who conducted the trial. Also, we see 12 months progression-free survival. It's near 76% versus 34% or 35% nearly. Overall survival was statistically or numerically in favor with trastuzumab deruxtecan, but not statistically significant, with 94% in 12 months overall survival. Progression-free survival benefits with trastuzumab deruxtecan is consistent across key subgroups, include those defined by HR status or prior anti-HER2 therapy, such as pertuzumab, or visceral disease, pre-mets, or number prior line of therapy. The response. I will not talk about numbers, but I will leave the graphs, such as BEST lesion reduction from baseline. From the graphs, the blue graph regarding trastuzumab deruxtecan with best percentage of best lesion reduction and or ORRs, overall relative response, is better in trastuzumab deruxtecan more than trastuzumab emtansine. I think that the graphs are comprehensive and conclude the lesion reduction from baseline. The adverse event of special interests is comparable. Most common adverse events associated with treatment discontinuation with trastuzumab deruxtecan, mainly pneumonitis, with trastuzumab emtansine, mainly thrombocytopenia. Most common associated with dose reduction, nausea and neutropenia in deruxtecan arm, and thrombocytopenia or liver enzymes elevation with emtansine arm. No grade 4 or 5 related interstitial lung disease or ejection fraction decrease. Also in deruxtecan arm, all ejection fraction decrease were asymptomatic and no cases of cardiac failure occurs. So that we can use it with particular safe more than the conventional anti-HER2 therapy, which regarded as cardiotoxic agents. After this, we will take drug-related adverse events, blood lymphatic disorders such as neutropenia, anemia, leukopenia, and thrombocytopenia. Both are comparable in both arms. Gastrointestinal were both also comparable. All of other side effects such as fatigue or liver enzymes increasing or decreased appetite or alopecia were also comparable. Conclusions of this clinical trial, phase 3 clinical trial, is that trastuzumab deruxtecan is statistically significant and clinically meaningful, has clinically meaningful progression-free survival benefit, with nearly 72% reduction in risk of progression than TDM1 in patients of previously treated HER2 metastatic breast cancer. Consistent benefit can seen across all patient subgroups, with overall relative response near 80% versus 35 or 34%. So overall survival data was numerically longer overall survival data with trastuzumab deruxtecan, but not statistically significant. Also, the safety profile comparable between two agents and drug-related adverse events. So the investigators conclude that the results support trastuzumab deruxtecan becoming the standard of care in second-line HER2-positive metastatic breast cancer. So this conclusion can change the practice, as the practice previously depend on first trastuzumab mainly, and then pertuzumab mainly, and then trastuzumab emtansine, and then after this promising trial, Destiny Breast, we lead to trastuzumab deruxtecan. So this can give us a clear message that phase 3 clinical trial can lead us to change of practice if the clinical benefits and the statistical significance can be seen in such these trials. My talk here about progression-free survivals, my talk here of overall responsive, the lesion volume reduction, and stuff like this. So I can conclude all the previous talk that cancer research start from one brick, the medical observation and basic research, and after this translational research to transform the basic research to the healthcare system or bedside. Then after we transform it to the bedside, we start to see the benefit of this observation for the patients, such as our trial here. We use two approved drugs, sorry, head to head to conclude or start a new rationale to use in metastatic breast cancer. So that conclude our talk that the cancer research is continuous cycle from basic, translational, clinical research, and then populational research, so that we can use to get modern inspection to make the treatment plans more reliable and more easy, and give more clinical benefits for the patient to help to cure or to detect or to screen or any other aspects that we use in clinical trial. I'm finished my talk, and thank you to have on board, and thank you for your times. Thank you very much for Dr. Nagam, and thank you for IVPN research team to this talk and to give me the chance to talk about this, and hope that talk, the conclusive talk, it will be helpful for you, and thank you very much. Waiting for your questions. Thank you.
Yeah, thank you, Dr. Mahrouz, for this informative presentation. Thank you so much. It's Fatima Asmail with you.
Yes.
Yeah. So thank you so much for the informative presentation. Actually, we learned a lot. It was very informative. I have actually one question. If the attendees, meanwhile, would like to submit their questions in the Q&A section.
Sure.
So what's the major safety alarming point that we need to monitor with such patient receiving this medication in specific?
For this, I think we need to monitor first of all the any condition regarding the lung, pneumonitis or interstitial lung disease, the ejection fraction, any case regarding the neutropenia mainly, or the neutropenia or the cardiac events such as ejection fraction reductions. I think that will be the more safety alarms that we will take in consideration for monitoring.
Okay, great. Is there any specific pre-medications the patient need to have prior receiving the treatment? Like nausea, vomiting,
sure.
or allergy?
Sure. The pre-medication is important in such cases. First, the allergy. We need to take pre-medication with Solicortev and diphenhydramine. The pre-medication with antiemetics regarding the moderately emetogenic or moderately emetogenic mainly, such as granistrone or palonostrone plus dexa, plus 8 milligram dexa in pre-medication. So that all the pre-medication needed. But regarding first time or the naive patient that will take such a treatment, I will focus mainly on the allergy, the drug-related allergy events. So I will give it in consideration in concurrent with the nausea and vomiting, but with less prescription.
Yes, great. Thank you so much, Dr. Mahrouz.
You're welcome.
Thank you so much. I would like to highlight to our attendees that if you can fill the certificate Zoom confirmation that's shared in the chat box. So we have Dr. Noor Burek. She's raising her hand. So please unmute yourself and ask your question, Dr. Noor.
Thank you, Dr. Mahrous, for your presentation. It was excellent. Thank you for clarifying the oncology research. And I like really the cancer tree. Working in epidemiology, I found this is very interesting because when we think about cancer research, we not only think about treatment, but also we need to think about prevention, care, screening, and other branches, not only pharmacology, but epidemiology. I like that you mentioned the physics, epidemiology, biology, and all of this. For the population research, I just want to raise one point that when we talk about epidemiology, we not only talk about the prevention or even assessment of the treatments from pharmacoepidemiology point of view, but also we can help in cure. This is just my, my comment. One more thing, we need to think really about when we talk about clinical trials or the treatments, the one that you just give us an example about, I would love to see who's like the funding, like this kind of research, like if you provide us in the references, when it comes to clinical trial and treatment, who's funding that specific research or that trial? Is that the pharmaceutical company, government, who like exactly the side of, just to, the conflict of interest, to be like for us as attendee or researcher, we need all the time